Hi everyone. In this video we're going to look at eukaryotic cell structure from AQA topic 2. So firstly we'll go over the structure and function of each of the organelles as listed in the specification, and then we'll have a look at specialised cells and how they're organised into tissues, organs and systems. So we'll start off with looking at the difference between eukaryotic and prokaryotic cells. These are the two main groups that you need to know about. So eukaryotic cells contain membrane-bound organelles. This just means that their organelles, or many of their organelles, are enclosed within a membrane. Prokaryotic cells do not contain membrane-bound organelles. They still have a cell surface membrane, but for example, they don't have a nucleus, whereas a eukaryotic cell does. So you will know eukaryotic cells best as plant and animal cells, but this group also includes fungi and protists. The prokaryotes you can think of as being bacteria, but there is a second group within the prokaryotes called archaea. They are unicellular, but they do have some differences between them and bacteria. So for example, their cell wall is made of a different substance. The first organelle we'll look at is the cell surface membrane, as this is an organelle that is present in all cells. So the cell surface membrane distinguishes the cell from the outside of the cell. So for example, viruses are not made of cells, so they don't have a cell surface membrane. It's made of lipids and proteins, and in fact the main component is a phospholipid bilayer, and proteins are embedded within this. You will look at the structure of the cell surface membrane in much more detail in a separate topic. So the membrane regulates the movement of substances into the cell and out of the cell, but it also has other roles. So some of the proteins, for example, may form receptors that are involved in cell recognition and signaling. So you'll look at some of that within the immune system. Some of the proteins may actually be enzymes too, so you'll come across this when you look at digestion. Next, we'll look at the nucleus, and in fact the nucleus is the big telltale sign of whether a cell is eukaryotic or prokaryotic, as the prokaryotes don't have these membrane-bound organelles, such as the nucleus. The nucleus, in fact, has two membranes, so we say it's surrounded by a double membrane that we call the nuclear envelope. This nuclear envelope contains nuclear pores, so these are little holes that the mRNA can leave the nucleus through. However, the DNA is too big and will stay within the nucleoplasm. So the DNA for most of the time does not exist as chromosomes, that's just when the cell is dividing. Most of the time in eukaryotes it exists as chromatin. So this is when it's slightly coiled up and wrapped around some proteins, and you will look at the structure of this a little bit later in the course. We also have a dense region within the nucleus that we call the nucleolus, and this is where we'll find a lot of ribosomal RNA because this is the site of ribosome synthesis. We've got another organelle that you should have already heard of, the mitochondria, and once again it's a membrane-bound organelle, and like the nucleus it still has a double membrane. This time the inner membrane is folded into structures called cristae, which increase its surface area. This is for some of the reactions that occur in aerobic respiration, so again you'll come on to this a little bit later. Mitochondria also contain their own DNA, so this exists within the inside of the inner membrane, which is an area we call the matrix. This DNA will contain the code for enzymes that are required in aerobic respiration and other proteins that are needed in those processes. So in order to make those proteins within the matrix, we also find ribosomes. Now we come on to chloroplasts, so you will know that they are only found within plant cells, but they are also found within some of the protists, so such as algae. They also contain chloroplasts so that they can photosynthesize. So once again, we've got a double membrane. Inside the inner membrane, we have a space called the stroma, and within that we find these thylakoid discs that are stacked into grana. One is a granum, multiple is grana, and these are linked by flat structures called lamellae. So once again, some of the reactions of photosynthesis will happen across that thylakoid membrane, and you will look at this again in topic 5. Some of the photosynthesis reactions occur within the stroma of the chloroplast. Again, the chloroplast contains its own DNA so that it can code for the proteins and enzymes that are required for photosynthesis. So we've got ribosomes there too for protein synthesis, and we'll also see lipid and starch granules as the food that is made in photosynthesis. The permanent vacuole is another membrane-bound organelle found within plant cells. This time, the membrane is referred to as the tonoplast, and inside the tonoplast we find cell sap. So this cell sap contains mineral ions, dissolved sugars, amino acids. So having a low water potential, it encourages water to move into the vacuole, which applies hydrostatic pressure on the cell and keeps the cell firm and turgid, and this supports the whole plant. The vacuole can also contain other substances, so for instance there may be chemicals within the vacuole to deter insects, or within flowers there may be pigmented chemicals to give the flower the petal colour. Some eukaryotic cells have a cell wall, which supports the cell and prevents any changes of shape. There are 2 types of cell wall that you need to be aware of within the eukaryotes. So firstly, you will be familiar that plant cells have a cell wall. You need to know that this is made of cellulose, and you will have looked at the structure of cellulose and how the microfibrils provide high tensile strength to resist the osmotic pressure. The second type is in fungi. This time the cell wall is made mainly of a substance called chitin. This is a carbohydrate. You don't need to know the specific structure of this, but it's another structural carbohydrate similar to cellulose. Now we come on to perhaps a new organelle, the endoplasmic reticulum. So there are 2 types of endoplasmic reticulum. First we'll look at the rough endoplasmic reticulum, and a little bit later we'll come back to it and look at the smooth endoplasmic reticulum. So the rough endoplasmic reticulum is usually found very close to the nucleus, and in fact it's another membrane-bound organelle, and that membrane is continuous with the nuclear envelope. The key feature of the rough endoplasmic reticulum is that it is studded with ribosomes because this is involved in protein synthesis. The membranes are flattened into sacs called cisternae, and once the polypeptide has been formed on the ribosome, the polypeptide will be folded into a protein and processed within the rough endoplasmic reticulum. So now we'll look in a little bit more detail about these ribosomes. So this is not membrane bound. In fact, it's a very, very tiny organelle, and it's made of ribosomal RNA and proteins. It consists of 2 subunits, so one is a small subunit, one is a large subunit. And these may be free within the cytoplasm or attached to the rough endoplasmic reticulum. It depends on which type of protein is being produced. So proteins that are used within the cytoplasm would be translated on the ribosomes that are free within the cytoplasm. If a protein is for use within the cell membrane, or it's going to be secreted, it's likely that it will need more processing, and so therefore it will be translated on a ribosome that is on the rough endoplasmic reticulum. So this, most importantly, is the site of protein synthesis. So another organelle that is involved within the secretion of proteins is the Golgi apparatus and the vesicles that are produced by it. So this is a series again of flattened membrane sacs that we call cisternae, and it forms a kind of cone shape. So that's the identifying feature here. Once a polypeptide has been produced on the ribosomes and processed in the endoplasmic reticulum, it's going to reach the Golgi apparatus for further processing and packaging. So, for example, a protein may become a glycoprotein by having a carbohydrate element added to it and then packaged into a vesicle, which will transport it to the cell membrane. So we've dealt with the rough endoplasmic reticulum and its role within protein synthesis, but now we come to the smooth endoplasmic reticulum. So this is a part of the endoplasmic reticulum that does not have any ribosomes on its surface. So again, it's membrane bound, but this time rather than flattened sacs, we see more of a tubular-like structures. And this smooth endoplasmic reticulum is involved in the production and storage of lipids and carbohydrates. The final organelle we need to be aware of for AQA is a lysosome. And this is in fact a specialised vesicle that specifically contains hydrolytic enzymes that you could refer to as lysozyme. So these are responsible for digesting invading cells, so they're very important within phagocytosis of the immune response, but they are also going to digest any worn out cell components so that the subunits could be recycled and reused in other molecules. So in the exam, you may be asked to interpret specialised cells, so how are they good at what they do? How is their structure suited to their function? So we find specialised cells in multicellular organisms. All the cells can't be the same because they're all fulfilling a different function, so they need to be structured slightly differently. So we're going to look at an example of the intestinal epithelial cell. So what we need to do is look at the image that we're given or the description and look at the organelles it contains. Think about the function and how the structure links to the function. So the intestinal epithelial cell, its main job is to absorb the products of digestion. So what is it in that structure that makes it useful for this function? So first of all, we can see at the top of the cell, we've got these extensions of the cell membrane called microvilli. So these are folds or extensions, and this increases the surface area for absorption to occur across. Secondly, what we can see is there are lots of mitochondria within the cytoplasm. Lots of mitochondria means lots of ATP is being produced, and so we now think, well, what could that be used for? So that's for energy, for transport across the membrane. The last concept to give a little bit of thought to is how these cells are organised. So firstly, similar specialised cells are grouped together to form tissues. So many epithelial cells will form epithelial tissue, and we can see all of those have got those microvilli structures on them. These epithelial cells combined with perhaps goblet cells that secrete mucus, and we can see some blood vessels there. So there's going to be blood tissue, there's lymphatic tissue. Those are going to be grouped together. We can see a structure called a villus there, and these are forming the small intestine. So they're forming an organ. So different tissues working together, forming an organ. The small intestine combined with the stomach and the liver and the pancreas and the esophagus and so on form the digestive system. So we've got these different organs making up an organ system. So that's it for eukaryotic cell structure. So make sure you're aware of the structure and function of all the organelles in this list. These are the ones that are stated within the specification. Have a little bit of practice of linking the structure of specialised cells to their function, and make sure you're aware of how cells are organised into tissues, organs and systems. In the next video, we'll look at prokaryotic cells and viruses.